Okay, let's unpack this. Imagine a super complex, lightning-fast communication network. It runs, well, everything in your body, from your deepest thoughts to the simplest twitch of a finger, even your breathing when you're not thinking about it.
That's your nervous system, the ultimate command center.
Absolutely. And today we're taking a deep dive into its incredible structure and how it actually works. We're using an excellent source, a chapter from Understanding Pathophysiology, the 7th edition.
Right, by Huether, McCants, Brashers, and Rote. And what's great about this chapter is how it systematically builds our understanding, layer by layer.
So what's our mission today?
Our mission is really to be your guides through this material. We'll walk you step by step through the major concepts, look at the mechanisms, and even touch on a few key clinical connections.
And we'll try to make it really clear, right? Like painting a picture with words, describing any figures or tables mentioned, so you can visualize these amazing systems even without the book in front of you.
Exactly, making complex terms accessible.
Perfect. So whether you're maybe a college student cramming for an exam or just someone really curious about the machinery inside you, get ready for some, hopefully, aha moments.
Let's start big picture.
Okay, the grand organization. How is this massive system structured?
Well, fundamentally, it's divided into two main parts, which helps break it down. First, you've got the central nervous system, the CNS.
That's the brain and spinal cord, right?
Exactly, the command hub, safely tucked away inside your skull and your vertebral column, your backbone.
Okay, central command. Then what's the other part?
That's the peripheral nervous system, or PNS. Think of this as all the wires and cables extending out from the CNS.
Like cranial nerves, spinal nerves?
Precisely. They reach literally every other part of your body. So CNS is the processing unit, PNS is all the input-output connections.
Got it. And within the PNS, how does information flow?
Great question. There are two main pathway types. You have afferent pathways. These are sensory messengers. They carry information from your body towards the CNS.
So like if I touch something hot.
Exactly. That heat sensation travels up an afferent pathway. Then you have the efferent pathways. These carry motor commands away from the CNS to your muscles and organs, telling them what to do.
Like pulling my hand away from that hot thing.
Precisely. It's a two-way street for information.
Okay, structural divide, clear. But functionally, what does the PNS do? How does that break down?
Functionally, the PNS has two big jobs. First, the somatic nervous system. This is your voluntary control system.
So deciding to move my arm, walk, talk.
That's it. It controls your skeletal muscles, things you consciously decide to do. Then there's the autonomic nervous system, the ANS. And this one's fascinating because it's completely involuntary.
The autopilot system.
Pretty much. It manages your internal environment, heart rate, digestion, breathing rate, blood pressure, all without you having to think about it.
And the ANS has divisions too, right?
It does. Two famous ones that often work in opposition. The sympathetic and the parasympathetic nervous systems. Think fight or flight versus rest and digest. We'll definitely get into their tug of war later.
Wow. Okay, it's amazing how much runs automatically. Now, let's zoom in. What are the actual building blocks, the cells doing all this work?
Right, the fundamental units. The stars are definitely the neurons. These are the primary communication cells. They're electrically excitable, designed to transmit and receive information incredibly fast.
What do they look like, if we could picture one?
Okay, imagine, like in figure 14.2b in the text, a central cell body, or soma, that's the neuron's metabolic center, keeping it alive. Branching off from it usually are these thin, tree-like fibers called dendrites.
Like little antennas.
Exactly. They bring impulses towards the cell body. Then there's typically one longer projection called an axon. This carries impulses away from the cell body.
Sending the message onward.
Right. And where the axon joins the cell body, there's a little cone-shaped region called the axon hillock. That's usually where the electrical signal, the action potential, gets started.
You mentioned speed. How do they achieve that incredible speed?
That's largely thanks to the myelin sheath. Many axons are wrapped in this fatty insulating layer. Think of it like the plastic coating on an electrical wire.
Okay, insulation.
Right. In the CNS, it's made by cells called oligodendrocytes. In the PNS, it's Schwann cells. But this myelin isn't one continuous sheath. There are tiny gaps.
Gaps, why?
Those gaps are called nodes of Ranvier, and they are absolutely critical. The electrical impulse doesn't just flow smoothly down the axon, it actually leaps from one node to the next.
It jumps.
It jumps. This process is called saltatory conduction, and it dramatically speeds up the signal transmission. miles per hour faster than unmyelinated axons.
Wow. So that explains why damage to myelin, like in multiple sclerosis you mentioned, is so devastating. The signals just slow right down.
Or stop altogether. It really highlights how vital that insulation is.
But neurons aren't the only cells involved, are they? There's a support crew.
Absolutely. You can't forget the neuroglia, sometimes called nerve glue. These non-neuronal cells make up about half the volume of your brain and spinal cord.
Half. What do they all do?
They're the unsung heroes. Take astrocytes. They provide structural support, help move nutrients to neurons. They're a crucial part of the blood-brain barrier that protects the brain. They even form scar tissue.
Dizzy cells.
Very. Then you have the oligodendrocytes and Schwann cells, the myelin makers we just discussed. And microglia, these are the brain's immune cells, like tiny janitors cleaning up debris and fighting infections.
So while neurons get the spotlight, the neuroglia are essential for everything to function.
Couldn't function without them. If you look at figure 14.3 or table 14.1 in the text, you really see the diversity of these support roles.
Okay, this raises a tough question then. What happens if these cells, especially the neurons, get injured?
Yeah.
Can they repair themselves?
That's tricky. Unlike many cells, mature neurons generally don't divide. So a significant injury can mean permanent loss of function.
So if an axon gets cut,
The part of the axon separated from the cell body will degenerate. It's called Wallerian degeneration. It swells up, the myelin breaks down, and eventually the axon segment disappears. It basically starves.
can it ever grow back?
In the peripheral nervous system, there's some limited potential for regeneration, especially in myelinated nerves. The Schwann cells can actually form a sort of tube, a guiding channel. Figure 14.4 shows this, that the axon might be able to regrow along.
Might be able to.
Yeah, it's not guaranteed. Depends on the injury. Type crushing is often better than a clean cut. How close it is to the cell body, inflammation, scarring, lots of factors. In the CNS, though, regeneration is very limited. Scar tissue formed by astrocytes gets in the way, and oligodendrocytes don't support regrowth like Schwann cells do.
So recovery is much harder in the brain and spinal cord.
Much harder, unfortunately. Closer the injury to the cell body, generally the worse the outcome.
Okay, here's where it gets really fascinating for me.
Mhm.
How do these individual neurons actually talk to each other? How does the signal jump from one to the next?
That critical communication happens at the synapse. Think of it as a specialized junction, a tiny gap between two neurons. They don't physically touch.
No touching. How does the signal cross?
Chemicals. You have the neuron sending the signal, the presynaptic neuron, and the one receiving it, the postsynaptic neuron. When the electrical impulse, the action potential, reaches the end of the presynaptic axon,
The axon terminal.
right, the synaptic knob, it triggers the release of chemical messengers called neurotransmitters from tiny storage sacs called vesicles.
Okay, chemicals released into the gap.
Into that gap, the synaptic cleft. They diffuse across and bind to specific receptors on the postsynaptic neuron's membrane, like a key fitting into a lock. Figure 14.5 illustrates this step-by-step chemical conduction beautifully.
And that binding triggers a new signal in the next neuron.
Exactly. It causes a change in the postsynaptic neuron, potentially starting a new electrical impulse there. It's how the message gets relayed.
And this happens billions of times all over the nervous system.
Constantly. And what's amazing is neuroplasticity, the brain's ability to change and adapt throughout life by reorganizing these pathways, forming new synapses. That's fundamental for learning, memory, even recovery after injury.
Incredible. So these neurotransmitters, they're the chemical language. Can you give us some key examples?
Absolutely. They're the messengers, and their balance is critical. Take acetylcholine. It can excite or inhibit, crucial for muscle contraction and memory. Low levels are implicated in Alzheimer's disease.
Okay. What else?
Norepinephrine, involved in alertness, attention, the fight or flight response. Cocaine and amphetamines actually work by increasing its effects. Serotonin is generally inhibitory, think mood, anxiety, sleep regulation.
Many common medications target serotonin, right?
Yes, absolutely. Then there's dopamine, mostly excitatory, vital for movement control and the brain's reward pathways. The loss of dopamine-producing neurons is the cause of Parkinson's disease.
Such specific roles. Any others?
A really important one is GABA, gamma-aminobutyric acid. It's the main inhibitory neurotransmitter in the brain. It helps calm neuronal activity. Drugs that boost GABA are used to treat things like epilepsy. Table 14.2 in the chapter lists many more, but these give you a sense of their diverse and vital functions.
It's an intricate chemical ballet. So we've covered the building blocks, the communication. Let's move up to the command center itself, the central nervous system. Starting with the boss, the brain.
Right, the brain. About 3 pounds of incredible complexity. It uses a massive amount of energy, 15 to 20% of your heart's output. It's the seat of reason, intellect, personality, mood, everything that makes you, you.
How does it develop? Is it complex from the start?
It starts simpler, embryonically, from 3 main parts: the forebrain, midbrain, and hindbrain. These then develop into the structures we recognize. Table 14.3 gives a nice overview.
So forebrain
The
becomes.
forebrain gives rise to the telencephalon, which becomes the large cerebral hemispheres. The wrinkly part we usually think of as the brain, and the diencephalon, containing the thalamus and hypothalamus deep inside.
Midbrain.
The midbrain stays relatively simple, acting as a key relay station. The hindbrain develops into the cerebellum, important for coordination, the pons, a bridge structure, and the medulla oblongata, controls vital functions.
And connecting all these.
Connecting the hemispheres, cerebellum, and spinal cord is the brainstem, which includes the midbrain, pons, and medulla. It's also home to the reticular formation, a network crucial for wakefulness and attention, the reticular activating system.
It's amazing how specialized different areas are.
Yeah.
But you mentioned they work as a network.
Absolutely. While we talk about functional localization, like Broca's area for speech production, the brain functions through incredibly complex, interconnected networks. Box 14.1 in the text touches on this field of connectomics, mapping these pathways. It's not just isolated regions doing their own thing.
Fascinating. Let's dive into the forebrain more, starting with those big cerebral hemispheres.
Okay. The outer layer is the cerebral cortex. That's gray matter, mostly neuron cell bodies. It's highly folded, with ridges called gyri and grooves called sulci, or deeper fissures,
Viola folds.
to increase the surface area. It packs vastly more processing power into the limited space of your skull. Beneath this gray cortex is the white matter, the myelinated axon pathways connecting everything.
And the hemispheres are divided into lobes, right? Like geographic region.
Exactly. Figure 14.8 shows this well. You have the frontal lobe at the front, your executive control center, goal setting, planning, short-term memory, inhibiting inappropriate behavior. It's all happening there.
And movement control.
Yes. The posterior part of the frontal lobe contains the primary motor area on the precentral gyrus. This is where voluntary movements originate. Remember the homunculus we mentioned? That map of body parts is right here. More cortex dedicated to fine control areas like hands and face. The frontal lobe also houses Broca's speech area, usually on the left, for the motor production of speech.
So damage there affects speaking ability.
Yes, expressive aphasia, difficulty forming words. Moving back, the parietal lobe receives and processes sensory input, touch, temperature, pain from the body. Its front edge, the postcentral gyrus, is the primary somatosensory area.
Occipital lobe.
At the very back, that's your occipital lobe, dedicated to processing visual information.
And the temporal lobe.
On the sides, beneath the temples, the temporal lobe handles auditory processing, contains the primary auditory cortex. Critically, it also includes Wernicke's area, essential for understanding language. Damage here causes receptive aphasia. You can hear words, but they don't make sense. It's also involved in memory and smell.
Are there any other lobes?
There's one hidden deep within, the insula. It processes sensory and emotional information, among other things, and connecting the two hemispheres, allowing them to communicate constantly, is the massive bundle of white matter called the corpus callosum, vital for coordination.
Okay, that's the cortex. What about structures deeper inside the forebrain?
Deep within, you find the basal ganglia. These are groups of subcortical nuclei like the caudate nucleus, putamen, globus pallidus, and the dopamine-producing substantia nigra. They're crucial for smoothing out voluntary movements and involved in cognitive and emotional functions too.
Is this where Parkinson's disease has its effect?
Yes, the loss of dopamine from the substantia nigra disrupts the basal ganglia circuitry, leading to the movement problems seen in Parkinson's. Huntington disease also involves the basal ganglia. These structures are part of the larger extrapyramidal system, which handles more involuntary reflexes and coordinated movement.
What about emotions and memory?
That brings us to the limbic system. It's not one structure, but a network, including parts like the amygdala, emotion, fear, hippocampus, memory formation, and hypothalamus. It's involved in primitive behaviors, emotion, motivation, mood, memory, and even smell.
And the diencephalon. You mentioned thalamus and hypothalamus.
Right, nestled deep between the hemispheres. The thalamus is like the brain's grand central relay station. Almost all sensory information going to the cortex passes through the thalamus for processing and routing. Below it is the hypothalamus. Tiny, but incredibly powerful.
Master regulator, you said.
Absolutely. It maintains internal balance, homeostasis, controls the autonomic nervous system, regulates body temperature, hunger, thirst, sleep-wake cycles, links the nervous system to the endocrine system, hormones, and influences emotional expression. Docs 14.2 lists its many crucial functions. It's vital.
Wow. Okay, moving down from the forebrain, the midbrain.
The midbrain, or mesencephalon, acts as a relay center for motor and sensory tracts. It has structures involved in visual and auditory reflexes, like automatically turning your head towards a sudden sound or light. The cerebral aqueduct, carrying CSF, runs through it.
And below that, the hindbrain.
Right. This includes the cerebellum, located at the back, underneath the occipital lobes. It's essential for coordinating voluntary movements, maintaining balance, and posture. It fine-tunes motor commands.
So smooth, coordinated actions rely on the cerebellum.
Heavily. Then there's the pons, acting as a bridge, transmitting information between the cerebellum and the brainstem, and between the brain hemispheres. And finally, the lowest part of the brainstem, connecting to the spinal cord, is the medulla oblongata.
Medulla. That controls vital functions, right?
Yes, absolutely critical. Heart rate, respiration, blood pressure, reflexes like coughing, swallowing, vomiting. These are all regulated by the medulla. It's also where most major motor pathways cross over to causation.
Ah, so that's why the left brain controls the right body and vice versa.
That's the spot where it happens, contralateral control.
Okay, we've toured the brain's command centers. How do those commands get out and sensations get in? Let's talk about the spinal cord, the information superhighway.
Exactly. The spinal cord connects the brain to the body. It handles reflexes on its own, integrates signals, and provides the pathways for motor commands going down and sensory information coming up. It starts at the medulla and runs down to about the first or second lumbar vertebra.
What does it look like inside?
If you took a cross-section, like in figure 14.12, you'd see an inner butterfly-shaped region of gray matter. This contains neuron cell bodies. The wings are called horns. The posterior or dorsal horn receives sensory input. The anterior or ventral horn contains motor neuron cell bodies whose axons go out to muscles. In some regions, there's a lateral horn for the autonomic nervous system.
And surrounding the butterfly.
That's the white matter, organized into columns or tracks. These are the myelinated axons carrying signals up, ascending sensory tracts, and down, descending motor tracts, the cord.
You mentioned reflexes earlier. How do they work at the spinal cord level?
Through reflex arcs. These are basic neural circuits. Figure 14.13 shows a simple one. A sensory receptor detects a stimulus, like heat, sends a signal via a sensory neuron into the dorsal horn. It might connect directly or via an interneuron to a motor neuron in the ventral horn, which sends a signal out to a muscle, causing a response, like pulling your hand away.
All happening within the cord super fast.
Before the brain even fully registers it sometimes, it's protective.
Let's talk about those motor neurons, upper and lower.
Right. Upper motor neurons, UMNs, have their cell bodies in the CNS, like the motor cortex. They control and modify the activity of lower motor neurons. Lower motor neurons, LMNs, have cell bodies in the brainstem or spinal cord ventral horn, and their axons extend out into the PNS to directly synapse on and control muscles.
So damage to each has different effects.
Yes. UMN damage often leads to spasticity and weakness, but some movement might remain or recover. LMN damage leads to flaccid paralysis and muscle atrophy as the muscle loses its direct nerve supply. Recovery depends on nerve regeneration, which is limited, as we discussed.
And a single LMN connects to multiple muscle fibers.
Yes, a single lower motor neuron and all the muscle fibers it innervates is called a motor unit. The connection point is the neuromuscular junction, shown in figure 14.14.
Okay, so how do the big motor commands travel down from the brain? The pathways.
The major pathway for precise, voluntary movements, especially of the limbs, is the lateral corticospinal tract, also called the pyramidal tract. As we said, these UMNs start in the motor cortex, descend, and crucially, most crossover, decussate, in the medulla to control the opposite side of the body. Figure 14.14a shows these tracts.
Are there other motor pathways?
Yes, the extrapyramidal tracts. These originate in the brainstem and are involved in things like maintaining posture, balance, muscle turn, and coordinating gross movements. Examples are the reticulospinal and vestibulospinal tracts.
Okay, that's motor output. What about sensory input coming up?
Sensory information ascends via specific pathways too, shown in figure 14.14. B, fine touch, pressure, two-point discrimination, proprioception, knowing where your limbs are. That detailed epicritic information travels up the posterior or dorsal column pathway. These neurons run all the way up the cord on the same side they entered, synapsing in the medulla.
That's the really long neuron pathway you mentioned earlier.
That's the one. Then, sensations like pain, temperature, and crude or vague touch protopathic information travel up the anterior and lateral spinothalamic tracts. What's key here is that these neurons cross over to the opposite side of the spinal cord shortly after entering and then ascend to the thalamus.
So sensory pathways cross too, but at different levels depending on the type of sensation.
Exactly. This precise routing is critical for pinpointing where sensations come from or where motor control originates.
It's incredibly organized. But none of this delicate machinery would work without protection and fuel.
Hmm.
Let's talk about the protective structures and blood supply.
Absolutely vital. First, the cranium, the skull. 8 bones fused together form a rigid vault protecting the brain.
And inside the skull.
We have the meninges, those 3 protective membranes we touched on earlier. Figure 14.4c shows them nicely. Outermost is the tough duramater. It actually has 2 layers in the skull, forming rigid partitions like the falx cerebri separating the hemispheres.
Then the arachnoid.
Yes, the arachnoid mater, a spongy web-like layer beneath the dura. And finally, adhering directly to the brain and spinal cord surface is the delicate pia mater.
And the spaces between them are important.
Critically important. Between the dura and the skull is the potential epidural space. Skull fractures can tear arteries here, causing dangerous epidural hematomas. Between the dura and arachnoid is the subdural space. Tearing the bridging veins here can cause subdural hematomas, often seen in falls, especially in the elderly.
And the CSF is where?
In the subarachnoid space, between the arachnoid and the pia mater, this is where that cerebrospinal fluid, CSF, circulates.
Remind us about CSF function.
It's a clear, colorless fluid that cushions the brain and spinal cord, acts like a shock absorber, prevents tugging on structures, and helps maintain the chemical environment. It's produced by specialized ependymal cells in the choroid plexuses, found within the brain's interconnected cavities, the ventricles, shown in blue in figure 14.16a.
How does it circulate?
It flows through the ventricles, then out into the subarachnoid space surrounding the entire brain and spinal cord. Figure 14.16b maps this flow. Eventually, it's reabsorbed back into the venous blood system through structures called arachnoid villi, which act like one-way valves. Table 14.4 shows its composition similar to plasma, but distinct.
Constant production, circulation, and absorption.
Got it.
What about spinal cord protection?
The vertebral column. 33 vertebrae stacked up cervical, thoracic, lumbar, sacral, coccygeal sections, figure 14.16. Between most vertebrae are the intervertebral discs.
Shock absorbers.
Exactly. They have a tough outer ring and a soft, gel-like center, the nucleus pulposus. If you're 14.18, they cushion impacts. But if that outer ring tears, the nucleus can bulge or rupture, pressing on nerves, a common cause of back pain.
Okay, physical protection covered. What about blood supply? The brain needs a lot of fuel.
An incredible amount. Around 20% of your total cardiac output goes to the brain. Blood flow is tightly regulated, a process called autoregulation. Carbon dioxide levels are a major factor. High CO2 causes vasodilation to increase flow.
Where does the blood come from?
Primarily from 2 pairs of arteries. The internal carotid arteries coming up the front of your neck and the vertebral arteries coming up through the vertebrae at the back, shown in figures 14.19 and 14.20a.
And they connect somehow.
Yes. Crucially, at the base of the brain, branches from these arteries join to form the circle of Willis, figure 14.20b. This is that vital backup system. It provides collateral circulation, meaning if one major vessel is blocked, blood might still reach brain tissue via alternative routes through the circle, a lifesaver.
Amazing design.
And from the circle.
Major arteries branch off to supply different brain regions, the anterior, middle, and posterior cerebral arteries. Figure 14.21 maps these territories. Table 14.5 links specific arteries to functions. For example, blockage of the middle cerebral artery often causes speech problems, aphasia, because it supplies those language areas.
How does blood get out of the brain? Through cerebral veins, which drain into large channels within the dura mater called dural sinuses. These then empty into the large internal jugular veins in your neck, figure 14.22. Proper venous drainage is crucial for managing intracranial pressure.
And one more barrier, the blood-brain barrier.
Right, the BBB. It's not a wall, but rather highly selective cellular structures. The endothelial cells lining brain capillaries have very tight junctions between them, much tighter than elsewhere in the body. Astrocytes and other cells contribute too. Figure 14.23 shows this.
So it controls what gets from blood into brain tissue.
Exactly. It inhibits many substances, protecting the brain's delicate environment, but allows essential things like oxygen and glucose through. It's a major hurdle for drug delivery to the brain. Some drugs cross easily, others not at all. Breakdown of the BBB can contribute to neuroinflammation.
Spinal cord blood supply.
It gets blood from branches of the vertebral arteries and the aorta. There are anterior and posterior spinal arteries running along its length, reinforced by segmental arteries, figure 14.24.
Incredible layers of protection and supply. Now let's move out to the peripheral nervous system. What makes up the PNS?
It's all the nerves outside the brain and spinal cord. So the 12 pairs of cranial nerves emerging from the brainstem and the 31 pairs of spinal nerves emerging from the spinal cord.
What's the structure of a peripheral nerve like?
It's bundles of individual axons, or dendrites, many wrapped in that myelin sheath from Schwann cells. These axons are grouped into bundles called fascicles, and the whole nerve is wrapped in connective tissue layers, figure 14.25b.
Okay, the spinal nerves, 31 pairs, what do they do?
They emerge between the vertebrae, cervical, thoracic, lumbar, sacral, coccygeal regions. They are mostly mixed nerves, meaning each one carries both sensory fibers coming in and motor fibers going out.
Do they just go straight to their target?
Often, the anterior branches, rami, of spinal nerves intermingle to form complex networks called plexuses, like the brachial plexus supplying the arm or the lumbosacral plexus supplying the leg. This allows fibers from multiple spinal levels to contribute to the innervation of a limb.
And dermatomes, you mentioned those earlier.
Right. A dermatome is the specific area of skin supplied by sensory fibers from a single spinal nerve root. Figure 14.25c shows the map. Testing sensation in different dermatomes is incredibly useful clinically for pinpointing the level of a spinal cord injury or nerve root compression.
Like a diagnostic map on the skin. What about the cranial nerves?
12 pairs originating directly from the brain and brainstem, figure 14.25a shows their attachments. Unlike spinal nerves, some are purely sensory, some purely motor, and many are mixed. Table 14.6 lists them all.
Any key examples?
Sure. Cranial nerve I, the olfactory nerve, is purely sensory for smell. CN2, the optic nerve, purely sensory for vision. CN3 oculomotor is mainly motor, controlling several eye muscles and pupil constriction.
What about a mixed one?
A big one is CNX, the vagus nerve. It's mixed, carrying motor signals to the palate, pharynx, larynx, and a huge parasympathetic component controlling heart rate, digestion, and functions of many abdominal organs. It wanders far down from the head.
It's amazing how precisely these nerves are mapped. Okay, finally, let's revisit the autonomic nervous system, the involuntary controller. How does it maintain that internal balance, that homeostasis?
Its main job is coordinating and maintaining a steady state in things you don't consciously control: cardiac muscle, smooth muscle, in organs, blood vessels, and glands. It does this via those two main divisions we introduced, sympathetic and parasympathetic. Figure 14.26 gives a great visual overview.
The sympathetic, remind us.
Fight or flight. It gets you ready for action, mobilizes energy reserves, responds to stress. Think increased heart rate, blood pressure, blood sugar, dilated airways, redirected blood flow to muscles. Its nerve pathways originate in the thoracic and lumbar spinal cord, T1L2.
And the parasympathetic.
Rest and digest, or rest and repose. It conserves and restores energy, slows heart rate, enhances digestion, constricts pupils, promotes relaxation. Its pathways originate from cranial nerves, like the vagus, and the sacral spinal cord.
How does the wiring differ from the somatic system?
A key difference, shown in figure 14.27, is the two-neuron chain. In the ANS efferent pathway, a preganglionic neuron starts in the CNS and synapses in a ganglion outside the CNS. Then a postganglionic neuron goes from the ganglion to the target organ. The somatic system uses just one motor neuron from CNS to muscle.
And different chemical messengers, neurotransmitters.
Yes, that's crucial too. All preganglionic neurons, both sympathetic and parasympathetic, release acetylcholine. Parasympathetic postganglionic neurons also release acetylcholine, cholinergic transmission. However, most sympathetic postganglionic neurons release norepinephrine, adrenergic transmission.
So different chemicals, different effects.
And different receptors on the target organs. Adrenergic receptors respond to norepinephrine, and epinephrine from the adrenal gland. There are alpha receptors, one usually excitatory, other two usually inhibitory, and beta receptors. Adrenaline mainly increases heart rate contractility, beta 2 often causes relaxation, like bronchodilation. Acetylcholine binds to cholinergic receptors. Table 14.7 details these actions, like how sympathetic stimulation speeds the heart, while parasympathetic slows it, cholinergic.
So the overall function relies on this balance.
Exactly. The sympathetic system tends to cause widespread, generalized responses gearing up the whole body. The parasympathetic system often acts more locally, fine-tuning specific functions for rest and recovery. This constant interplay, often antagonistic, allows for incredibly precise regulation. Think about how sympathetic tone constantly maintains some level of blood vessel constriction. Vasomotor tone, figure 14.28a.
It's a dynamic balance act happening constantly. Now, as we wrap up, let's touch on geriatric considerations. How does aging impact this incredibly complex system?
Well, some changes are common, though highly variable between individuals. Structurally, there can be a slight decrease in brain weight and size, particularly in frontal areas, and ventricles might enlarge. Meninges can become more fibrous.
What about at the cellular level?
We might see some decrease in neuron numbers, though the link to functional decline isn't always direct. Decreased myelin, deposits of lipofuscin, an aging pigment, fewer synaptic connections, and the potential development of neurofibrillary tangles, hallmarks of Alzheimer's, can occur. Neurotransmitter balance can also shift.
And blood vessels.
Arterial atherosclerosis, hardening of arteries, can affect brain blood flow. The blood-brain barrier might become slightly more permeable, and vascular density could decrease.
How do these translate functionally?
Functionally, older adults may experience slower reflexes, declines in taste and smell, reduced vibratory sense, vision changes like needing more light, alterations in gait and posture, more fragmented sleep, and sometimes memory and cognitive impairments.
A reminder that even this intricate system changes over time.
Indeed, though it emphasizes the importance of maintaining overall health to support neurological function throughout life.
Okay, let's try to summarize this incredible journey. We've really covered a lot of ground.
We have. We started with the big picture, the CNS, brain and spinal cord as command central, and the PNS nerves as the communication network reaching everywhere else. We looked at afferent sensory in and efferent motor out pathways.
Then we dove into the building blocks. The amazing neurons with their dendrites, cell bodies, and axons sped up by myelin sheaths. And their essential support crew, the Neuroglia, doing everything from making myelin to fighting infections.
We saw how neurons communicate chemically across synapses using neurotransmitters like acetylcholine, dopamine, serotonin, and GABA, and how nerve injury, especially in the CNS, can be difficult to repair.
Then the grand tour of the brain. The forebrain with its complex cortex, frontal, parietal, temporal, occipital lobes, basal ganglia for movement, limbic system for emotion memory, and the vital thalamus and hypothalamus.
Followed by the midbrain relay station and the hindbrain's cerebellum for coordination, pons as a bridge, and the medulla controlling vital reflexes and marking the spot where motor pathways cross over.
We mapped the spinal cord's gray matter butterfly, and white matter tracks the ascending sensory pathways, like posterior columns and spinothalamic, and descending motor pathways like corticospinal, and those quick reflex arcs.
We can't forget the protective layers, the skull, the meninges, dura, arachnoid, pia, the cushioning CSF circulating through ventricles, and the subarachnoid space, the vertebral column shielding the spinal cord.
And the critical blood supply via carotid and vertebral arteries, the circle of Willis safety net, and the selective blood-brain barrier.
Then out to the periphery, the spinal nerves forming plexuses and mapping onto dermatomes, and the 12 pairs of cranial nerves with their diverse functions.
Finally, the autonomic nervous system, the involuntary master controller, balancing the fight-or-flight sympathetic division against the rest and digest parasympathetic division, using that two-neuron chain and specific neurotransmitters.
And we briefly touched on how aging can impact these structures and functions. It's a truly vast and intricate system.
It really is. So here's something to think about. Given this incredible complexity and how interconnected everything is, how might even a seemingly small problem in one specific area, say one type of neurotransmitter or one small pathway, potentially cause ripple effects, maybe widespread symptoms throughout the rest of the body?
That's a great point to ponder. It really underscores the delicate balance and integration of the nervous system. Its resilience is amazing, but sometimes its interconnectedness means disruptions can spread.
Absolutely. Well, thank you for joining us on this deep dive into the nervous system, guided by that excellent chapter from Huether and McCants. We hope you feel much more informed and maybe just a little more awestruck by the incredible biological machinery within you.
We certainly enjoyed exploring it. Thanks for listening.
We love digging into these topics with you, and we look forward to our next deep dive.